we are going to start discussing the SARS-CoV-2 vaccination and the strategies for the vaccinations. So let us start. This is a very important topic. I have been getting a lots of questions about it, that when will a vaccine be available? How are the vaccines going to be created? What will be the efficacy of the vaccine? How long will it help us? And so on. So realize this, that this is a new virus or new strain of a virus. We do not know yet. It has not spent enough time with us that we know how long the immunity would last, how fast the mutations would occur, what kind of mutations these will be, and would that cause the vaccines to become not capable or stay relevant. But while discussing the vaccines, I would like to talk about one thing, and that is, when we make a vaccine, we usually make the vaccine against a very functional part of the virus or pathogen, which that functional part gives it the basic properties of being that pathogen. So if in some cases the virus has mutated away from that functional part, that would simply mean that the virus is not functioning the way we thought it would, and the virus is not the same virus anymore. And if that happens, maybe the virus would become benign, or if that happens, maybe the virus would have a different disease. So as long as the virus is going to cause us a disease of the kind, for example, COVID-19 in SARS-CoV-2 case, the virus structure has to stay same to continue to cause the same disease, and the vaccine would continue to work against that. So that is the basic idea. Walaikum assalam. Welcome. And welcome. So let's start. Today's discussion is really fascinating, and this discussion is going to be very important for you to understand how the vaccines are going to work. So let us start the discussion. I'm going to share my screen, and we would start the discussion about it. So look, the vaccines, the question is, what is the purpose of the vaccine? Purpose of the vaccine. Why are we making a vaccine? How will it help us? So I'm going to go over this very quickly. Remember that I've been discussing this. Let's say this is a coronavirus, right? In the coronaviruses, let's say SARS-CoV-2, I'm talking about the purpose of the vaccine. Why do we make a vaccine? What will the vaccine do? So thank you very much, guys. Thank you for joining. Good evening or good morning to every one of you. So here is the coronavirus. What happens is, we all know this, that the coronavirus binds to ACE2 receptors on our cells, correct? ACE2 receptors. The binding protein is called spike protein, or it's actually a glycoprotein, spike protein or S protein. Keep this S protein in your mind, because the vaccines are targeting this S protein. So first thing to keep in mind while you're looking at thinking about the vaccines, that the vaccine most of the vaccines are targeting the S protein, number one. S protein, number two, is the one that connects with the ACE2 receptor on our host cell. So this is our host cell. That means some cell, for example, respiratory epithelium of human beings. And this combination allows the virus to enter the cell and then replicate and become more cells. So we've done that discussion before. The second part, so keep S protein in mind. Keep ACE2 in mind, because the vaccination is going to be targeting these areas. The second thing to keep in mind is the immune response. And I have done this almost in all of my lectures. So I'm just going to quickly talk about what are we trying to do with the vaccination. What will be the vaccination helping us with? So here is what happens. Look, let's say the pathogen comes in. This is the coronavirus. Our innate arm, our macrophage, takes it up, takes it inside and breaks it down into smaller pieces. On the macrophage, imagine it presents the part of the S protein. I'm making the S protein like this. This is the viral S protein that the macrophage ate up the virus. It broke it up into smaller pieces. Those smaller pieces are now displaying outside of the macrophage on MHC2 molecule. We know this. We have been discussing this all along. Then what happens is a naive T cell connects with the T cell receptor with this macrophage. There are co-stimulations. There are other proteins that engage. The end result is that the naive T cell and macrophage are now connected with the antigen presented, and naive T cell is going to become active. Now, what it is going to do is that if there is interleukin 4 available, then the naive T cell will go the route of T helper 2 cells. And I'm going fast on this one because we have done this before. It will become a T helper 2 cell, which will then bind with a B cell, which is a white blood cell, which is a lymphocyte. Both are the same thing. It's a lymphocyte. It is also part of acquired immunity, acquired arm of our immune system. White blood cell, lymphocyte, or acquired arm. Now, remember this, that when a virus enters our cell, enters our body, even B cell will have it captured on its surface, on the B cell receptors. So now, not all the B cells will be capturing the same antigen. Many B cells would capture a different kind of antigens. We have talked about this, that the receptors on the T cells and B cells are of various kinds. Their binding sites are different. So let's say this is a B cell that has a binding site which can connect with the S protein of the virus. So now this B cell is holding on to the S protein. The B cell is then now connected with the T helper 2 cell that has become active. This helper 2 cell is now going to connect with the B cell plus release interleukin 4 and interleukin 5. This will activate the B cell. An active B cell becomes a plasma cell. What is a plasma cell? It is an active B cell that is actively making antibodies which it would release. Antibodies. Good so far? This mechanism is what we want to do with our vaccine. What are we trying to do? We are trying to make our immune system recognize that there can be an antigen of this S protein type. Correct? This is the S protein. Our purpose is to make our immune system recognize the S proteins in the future, and then when the actual virus will come in, they would attack it. So this is the point of the vaccination. So what is the vaccine eventually going to do? If you keep this picture in your mind, a purpose of the vaccine is to introduce the pathogen in our body in a way that it does not harm us, but in a way that our immune system can recognize the parts of the pathogen, become prepared, and when the actual pathogen comes in, attack it fast and kill it. Welcome, Salam. So does this make sense? Now the question is, and Christina, I'll look at your question in a second. The question is, well, when the virus comes into our cell, when we catch the virus, our immune system is going to do this anyways. Then why do we need a vaccine to do this? The difference is that if I catch the virus today, it can take me anywhere from 7 days to 14 days for my immune system to go through this whole process that I just showed you and to become active. And the virus, especially SARS-CoV-2, can so rapidly grow in our body, replicate in our body in number, and cause damage that before 7 to 10 days, it might even kill us. It might cause mild disease or it might cause severe disease. So we cannot wait for our immune system to respond. And in some people, the immune system is not strong enough to respond. So what we do is we go to our immune system and we say, hey, immune system, here is how the virus looks like. Get ready now before the virus comes in. And when the virus would come in, attack it. That is the point of the vaccine. Vaccine's point is not that vaccine is sitting in our body and it would not let the pathogen come in. Vaccine's point is to tell our immune system to become ready for a special kind of pathogen or antigen or foreign material. And when that material comes in, it would cause, the immune system will cause the reaction. So one second, I'm going to answer those questions. So how will this in future cause the reaction? So here is what happens. These active cells, B cells, some copies of the B cells that have now recognized the virus protein, they are saved as memory cells. They are asked to go to sleep. So they are sleeping. They're sleeping. Why? And they still have their hands out. They still have their receptor ready for connecting with the S protein, but they're sleeping. There are tons of them. There are many of them that are just sleeping. These are memory cells. So we have made our immune system recognize the virus protein. Then we have made it to allow some cells to go to sleep or become memory cells. Now, when in the future, when the virus comes in, let's say this is the virus comes in and virus has the S protein on it. And if that virus comes near this B cell, which was sleeping, B cell is going to open their eyes right away and say, okay, my time to start working, and immediately start producing the immunoglobulins, which will go and attack the virus and help neutralize and clear it. Right? So this is the important thing. What are we doing? What is the purpose of the vaccine? The purpose of the vaccine is to allow us to make memory cells which against that particular pathogen. Yes, absolutely. So Justin responded and Justin said, just like a memory bank. Absolutely, Justin. So Patho is right here. I've been doing this discussion many times before. So now there is a question, will the vaccine be sufficient for both viral variants SNL? So it would depend. So that is a question, that can we make the vaccine in a way that can target both of the types or strains. So now let me show you one more thing. This virus is mutating, and how are we tracking the mutation? Let me show you. So here, if you look at this site here, this site is called nextstrain.org. There they are tracking the mutations in SARS-CoV-2 virus. Once again, please keep in your mind. If the virus has mutated sufficiently that it cannot cause that disease again, then the virus is useless to us. We don't care for it. If the virus is still continuing to cause the disease, then it is the same virus. Maybe it still has the mutations, but these mutations do not take the functional part of the virus away. So if you see here, this virus had started from, let's say, China, somewhere over here, and then it went into various countries, and the color changes means that the virus kept changing. And if you click on any one of these things, for example, when I click here, it says no nucleotide mutation. So that means the virus changed, but the nucleotide of the virus did not change too much. The bases did not change. Here, if I click here, you can see that it is saying nucleotide mutation, amino acid mutation. So here there is a mutation in which there is nucleotide bases are changed and amino acids are changed. So the virus genome is mutating, and we are tracking that mutation. And ideally, vaccines should be able to still work against the core part of the virus, the functional part of the virus. Good. So the discussion so far, what I would like you to have in your mind, S protein is an important protein on the virus against which we are trying to make our immune system ready, one. Second, S protein works with ACE2 receptors on our cells, and that is what causes the virus to become internalized. So if somehow we can take care of the S protein, then the virus cannot bind to us and cannot go in the cells, plus we can kill the virus. And again, I know that some of the people put the comment out on YouTube that I should not use the word kill for a virus. WHO uses the word kills, hospitals use the word kill. So I'm just going to continue using that. Yes, it can be inactivated or reduced in viability, but I will use the word kill here. So these are the two things. Third thing to remember is the point of a vaccine is to make our immune system recognize some part of the virus so that and become ready for it within a few days, so that when the actual virus comes in, immune system can attack it within 1 or 2 days instead of 7 or 10 days, during which at least SARS-CoV-2 very rapidly destroys our cells. Good? Any issues so far? Because after this, I'm going to go into the vaccines strategies. Okay, so I am going to assume that we are good. Now let's continue towards the vaccine strategies. So I'm going to go back to my board. So let's look at the vaccine strategies. Vaccines. Look, vaccines, I will put them in two basic categories: traditional vaccines and modern vaccines. And this modern vaccine is an interesting one. I'll show you how they're working. The traditional vaccines can then further divided into attenuated vaccine made up of live attenuated pathogen, and we'll discuss what that is. Let's call it A. Another strategy is to give the killed, again people are going to say, why did you use the word killed, inactivated virus or pathogen. That is B strategy. Third strategy is to do a recombinant protein manufacturing, and I'll explain what that is, C. And there can also be recombinant vector for vaccines. And again, I'll explain what these are. Let's call them D. And finally, modern ones are E. So there are about 5 ways we can make vaccines. Keep in mind, we are talking about creating a vaccine primarily against the S protein. So let's start. I'm going to go one by one. So I'm going to start with A. What is a vaccine which is called attenuated vaccine? Or it is more correctly called live attenuated vaccine. Here is what happens. You take a pathogen, let's say coronavirus, and that coronavirus is human in humans, right? So you take a coronavirus from a human. You take that and put that in a culture where we have fibroblast cells. So let's say this is a fibroblast cell from a chicken. So when we put a lot of viruses in this environment with lots of fibroblast cells, because these are chicken fibroblast cells and the virus is coming from human, virus may not be able to enter this chicken cell. So what we do is we wait for the virus to mutate. And when it mutates, maybe one or two viruses will be able to enter the chicken cell and go in there. Then what we do is we take those cells, those viruses that have entered the fibroblast of the chicken cell, we bring them into another culture where there are chicken cells, fibroblasts again over there, and we allow this changed virus, mutated virus, to once again infect these cells. Now this virus is going to mutate a little more and going to infect even faster or with more ease and replicate with more efficiency. But look at what is happening. As we are moving towards this direction, the virus is now becoming from a human species, it is becoming a chicken species. So if you keep doing it 80 times, 100 times, then the virus would become a species for the chicken. So let's say this is the fibroblast. After 100 times of the infections and mutations, this virus is now changed. How is it changed? It is able to get into a chicken cell and infect that. And the other important thing is now this virus cannot replicate easily in humans because it has gone 100 steps away in mutation from the human strain. Now if we take this virus and put that into a human cell, this new virus, this changed virus, this virus will not be able to cause disease and replication, and it will not be able to replicate easily in humans. Now what will happen is when it ends up in the human cell, so let's say here is a human cell, this is now a attenuated virus because we taught it to work only in the chickens and not in the humans. Now human cell is going to present that, as I showed above, present that as an antigen. Our immune system would come and connect with that, and then the immune response will come in. Good. This immune response is against the chicken virus, but because the structures are very similar to human virus, if the actual human virus is now going to come in to this cell, our immune system would attack that as well. So this is our first mechanism for vaccination. What is the mechanism? This is called the live attenuated vaccine. What we do is we take a human virus, for example, you take SARS-CoV-2, you put that in a dish with the chicken fibroblast cells. The virus is not going to go into the chicken cells because it does not know them, but you keep asking it to become, let it mutate to the point where it becomes, it is able to go into the cell of a chicken. Then you keep repeating those cycles 80 or 100 times that the virus becomes very familiar with the chicken and it loses its capability to replicate in humans. Then you take that virus, it is now attenuated, it is a weak virus now, you put it in the human cell and the human immune system will become active. Once the human immune system is active, when the actual virus will come in, then the human immune system would respond to that. So again, as I said, the basic point of doing this was to train our immune system to recognize a pathogen. Is this clear? This is called the live attenuated virus vaccine. Clear? Second is the killed or inactivated virus. How does that work? Very simply, instead of attenuating the virus, you take a virus, you treat it with some chemical substances, you maybe heat it as well, and the result is that the virus structure is still there, but virus has become broken and it has become coagulated and it is not really able to do any replication. You take this virus as a vaccine and you inject that into humans or whatever is the host, and human immune system, so let's say this is the human cell, that virus will enter the human cell, immune system, as I showed above, immune system would respond, immune system would learn how to work with this. So because this is a dead virus or killed virus or inactive virus, immune system has become trained, but virus did not do any damage, no damage. So now if the actual virus comes in, our immune system is already trained and it would attack it. Good. This is the killed virus. Any issue with this mechanism? So yeah, Justin, exactly, denatured. So Justin said it is denatured. Correct. It is denatured. Thank you, guys. So again, the point, so second type of vaccines are with the inactivated vaccines. Third type. What are the third type here? I said, so the third is the recombinant, and I'm going to talk about both of the recombinant together. Recombinant proteins are very simple. What you do is, so recombinant vaccines. Long lecture, if I can talk about an hour for the recombinant, but let's just go through this very quickly. What we do in the recombinant is that we take the viral RNA. So let's say the SARS-CoV-2 virus, its gene looks like this, its RNA looks like this. It has open reading frame 1A, then open reading frame 1B. This is the RNA structure. Then if you can remember semen, the semen from ejaculation. So S protein, E protein gene, M protein gene, and then N protein gene. There are many other genes included, but this is the basic structure which we are interested in for the RNA. In this structure, we are interested in this S protein gene. What we want to do is, we want to take this gene out, this part of the RNA. We want to use this to make lots of S proteins. S proteins look like something like this. Remember, I told you in the beginning of the lecture that please keep the S protein in your mind. This is the spike protein that binds with the ACE2 receptors on our body cells, and these receptors allow this protein and virus to be internalized. So that protein has an S protein gene. We want to take that gene from the virus. We want to use it to make more S proteins. So there is no virus. We just want to make S proteins. So lots and lots of S proteins. And then we want to inject these S proteins into our body. So let's say human being here. Let's see if I can make a human being. So here is a human being. Here is his head. So we inject the S proteins into that person, and the person's, of course, the cells which would take up the S proteins, they would either show that on MHC1, or if these are antigen presenting cells, then MHC2, and the immune system will become active. Immune system. And I have done the immune system diagram above. Immune system will become active. But remember, this is only S protein. S protein is not going to cause any damage. But because it is made the immune system active, when the actual virus is going to come in, the dangerous one, the active one, the whole virus, immune system would attack it. Because immune system would recognize the S proteins. This is how the recombinant technology works. The question is, how do we, hey, Visaris, Alexandros, how are you? The question is, how do we make these S proteins? How does this happen? So I'm going to quickly show you how recombinant technology works. So here is what happens. We take the virus RNA, the sRNA, we take this out, we take a plasmid is kind of a DNA. We take the RNA piece for S protein, we create a complement DNA and then a DNA from it. Then we take a plasmid is a circular DNA. we use enzymes that are called restriction enzymes. Restriction enzymes. Restriction enzymes, they act like scissors and they can cut the DNAs. So they act like scissors and they can cut the DNA. So we tell the restriction enzyme the pattern of the RNA pieces that we want to cut between, and the restriction enzyme will bring this part of the RNA out. Then we make the DNA from that. Then we have another set of enzymes called ligases. We give these ligases and this, so let's say this is the RNA for S protein. We give it to ligases and ligases would paste that RNA's DNA complement into the DNA, we form the DNA from it, and that is pasted in the plasmid. Good so far? So we've taken the RNA from the SARS-CoV-2, the spike protein RNA, then we converted that into a complementary DNA, then we have connected that DNA into a plasmid. This plasmid also has an antibiotic resistant gene in it as well. Then we take this plasmid and put that into a bacteria. So let's say this is a bacteria. We put this plasmid in this bacteria as well. Do you know how the plasmid goes into the bacteria? It is very funny. You put the plasmid near the bacteria. This is the plasmid. You put this near the bacteria and you heat it up. When you heat the solution, bacteria somehow, we do not know what is the mechanism, but bacteria would eat up the plasmid. So as soon as you give the bacteria some heat, bacteria says, oh, yummy, I'm going to start eating things. And it eats up the plasmid and takes it in. When the plasmid goes into the bacteria, now you culture those bacteria in a nutritional environment with the nutrients. You also put antibiotics so that other bacteria do not grow in there. Because this bacteria has an antibiotic resistant gene in it, I'm going very fast, antibiotic resistant gene, that would allow only this bacteria to live, and other bacteria that are here, they would die. So this way we now have isolated the bacteria that has the plasmid in it, and these bacteria are now going to make copies of themselves. When they are making copies of themselves, we have tons of bacteria. And when this plasmid on those bacteria will work, what would it do? It would produce S proteins. Right? So this is a technology called recombinant technology, where we give the viral gene to a bacteria, and when bacteria grows and multiplies and works, it creates the S proteins. Now we take all of those S proteins that bacteria are making. This is called recombinant because we have recombined bacteria's gene with the RNA gene. We take these S proteins, we inject that into a host. Host cell would take up the S proteins, both normal cell that would present it on MHC1 and antigen presenting cell like macrophages, APC, that would present it on MHC2. That would cause the immune system to become active, as I did the diagram above. Now when the actual virus will come in, that would have the S proteins on it, our immune system would very quickly recognize it and attack it. So this is the recombinant technology. Any issue with this one? Any issue with this one? Thank you, Justin. Yes, it needs heating. We heat the bacteria with the plasmid and bacteria would eat up the plasmid. It would say, oh, yummy, I'm hot, I'm going to eat something, and it's going to become hungry and eat it. Okay, so I hope that this is also clear that how a recombinant technology works. Now, there is another way of working with the recombinant technologies, and that is, this is the last way of working with the recombinant technology, and that is you take a bacteria, let's say a bacteria that is not harmful to us, for example, BCG bacteria, or you take a virus that is not harmful to us, for example, adenovirus. Adenovirus. Adenovirus, when it goes into our body, it kind of not replicate a lot. Same thing for the BCG bacteria. You paste the plasmid into these bacteria with the genes for S protein, or in this virus with the gene for the S protein. Then we inject those back into human. This is human for me. Maybe this is me. You inject it into the human, and what happens is, of course, human immune system will become active. Bacteria would make the S proteins as well, which are for the virus. Immune system becomes active. It becomes trained. And when the actual virus will come in, guess what will happen? The immune system would recognize the S proteins or even other parts and attack it. Good? Good. So that is another way of doing recombinant therapy. Here we have given attenuated pathogens directly into the body. This is BCG or adenovirus. Any issue with this? So these are the traditional technologies. Traditional technologies are what? Number one, we can take up a pathogen. So if I look at here, take up a pathogen, inject the RNA from it into a virus or a bacteria like BCG or adenovirus, give that virus or bacteria into our body. The virus or bacteria would start replicating a little bit and make our immune system active against the S proteins. Immune system will become trained. In the future, it comes in like SARS-CoV-2, we'll attack it and kill it. That is one way. Second way is you take the RNA gene from the SARS-CoV-2 for S protein, put that into a bacteria, culture that bacteria, grow it again and again to the point that the bacteria start making S proteins in big volumes. Take those S proteins, inject them in the body, our immune system will react and become trained. When the actual SARS-CoV come in, we will attack it and kill it. Third is take the virus itself, chemically treat it, make it inactive, inject it into our body. It is a dead virus. It cannot do anything, but our immune system will become trained. When the actual virus comes in, we're going to kill it. Fourth, take the virus and then do what? Attenuate the virus. Put it in the chicken cells and keep making it live in the chicken cells and become used to chicken cells and become mutated to work with the chicken cells. When it has mutated about 100 times, then you take it from the chicken cells and inject it in the humans. Now the virus does not replicate well in the humans, but our immune system will become active and trained and memory will develop. When the actual virus comes in, we'll attack it and kill it. Good. So no, actually we will have a vaccine for the SARS-CoV. So if we are good so far, now I'm going to talk about the modern medicine that is being made, modern vaccine that is made. That is the last topic for today. So let's talk about the modern way. So there is a company in Germany, I believe its name is CureVac. CureVac. I have their website here as well. So if you look here, this is the CureVac company. This is their website for how they're making COVID-19 vaccine. So I'm going to show you this is a diagram of the S protein. This is the S protein on the virus against which we are trying to make vaccines. So now let's see what CureVac is doing. And I'm sure that there are 35 companies that are working on vaccines right now. They may be working on various types of vaccines. As I mentioned, I've already mentioned 4 types. I'm going to mention the last type, which CureVac is. It is more exciting for me to talk about it. It is more modern technology. Let's see how does this work. They call it mRNA vaccine. And it is being made. There are vaccines that are ready for human trials even now. It's just that vaccine trial needs, do you know what we need to do? First, we have to make a vaccine and try it on animals. So I'm going to try to make a little mouse here. If I don't make a good, this is a mouse. This is a mouse. So this is a mouse. So first we have to try it on animals. If it succeeds there, then we try it on a limited number of human beings, volunteers. If it succeeds there, then we try it on a larger group of human beings, maybe in a community where the infection is spreading. If it becomes successful there, then we give it in a larger population. So this is how these works. And this in the past used to take like 10 years to develop and work. Now they're saying that by this summer or by the early next year, we'll have the vaccine. So vaccines are coming up fast. Let's talk about the messenger RNA vaccine. It is a beautiful way of making the vaccine. I show on screen, please. Sure, you are on the screen now. All right, let's do it. Oh, sorry. So here is the screen. So I was talking about sorry, let me go back to screen here. So I was talking about the more modern messenger RNA vaccine. Vaccine, when you make, you have to try it on animals, then on some humans, then more humans, then a lot more humans. This can take a lot of time. So let's make the messenger RNA vaccine. Beautiful technology. Here is what we do. So let's take the virus SARS-CoV-2 RNA. So open reading frame 1A, open reading frame 1B, S protein, remember semen, E protein, M protein, N protein, genes. These are genes. And there are many other genes intervening. I'm just talking about the more important genes. So what they're doing is they're taking S protein RNA out of the virus RNA. So they're taking the S protein RNA out. Then what they're doing is they're creating a complementary messenger RNA from it. So what does that mean? A messenger RNA has a head and a tail. The head is usually AUG, that is called a start codon. And the tail is usually a poly-A tail. So about 6 A amino acids here. So they take the S protein gene, they convert it into a messenger RNA with the proper structure. Then they wrap this messenger RNA into a lipid nanoparticle. What is that? That is cell membrane, you can say. So they're making it right now. Somebody is saying, when would they make it? They're already making it. It's just that they need to do the testing. So this is a membrane. They take this messenger RNA they made and put it in here. Now this lipid nanoparticle, these things are created in millions and billions. Then they inject it into human beings. So now what happens is, check this out, it's beautiful. So let's take a human cell. So let's say this is a cell. This lipid nanoparticle allows the particle to enter the cell. Remember, it is lipid. Our cell membrane does not allow everything to get into it. But lipid particles can enter the cell membrane because cell membrane itself is phospholipids. So now this lipid particle with the messenger RNA in it is taken up by the cell. If we gave the messenger RNA alone, it will not be taken up by the cell. It would already be destroyed by our cell's enzymes or outside enzymes. Even when it would try to get into the cell, it cannot because cell would not allow it. So we kind of wrap this thing in a lipid particle as a vehicle to allow it to enter. So once it's inside the cell, the messenger RNA would come out, and it will be used like a normal messenger RNA to make S proteins. And the cells are now going to have a bunch of S proteins in them. It's a beautiful mechanism. It would just use cells machinery to make S proteins. These S proteins are going to be now presented on MHC1, or they will be presented on MHC2, as I've been discussing again and again. That would cause the immune system to become trained, and then when the actual virus comes in, which has these proteins on it, immune system would attack it. This is the messenger RNA technology. So I hope this is clear. How does this work? Messenger RNA technology, you take a piece of RNA from the virus, you make a messenger RNA from it, you wrap that into a lipid particle so the RNA can enter a cell. When the lipid particle is introduced and it goes into the cell, cell can now show it on MHC1 or MHC2, and then immune system becomes trained. And when the actual virus comes in, our body is going to just attack it and kill it right away. That is how these vaccines work. The vaccines, many companies are now saying that the vaccines are ready. They're ready for the trials. But of course, we have to understand if the vaccine is efficacious or not, if vaccine is going to cause damage, how well the vaccine would work, if the mutations are going to cause an issue. So these, all the things are still on the table, but vaccines are available. There are 35 companies that are making it. Many companies have said that we are ready for human trials as well. At least in the US, it is going to be difficult to start human trial right away. So maybe other countries would allow those trials. I do not know, but we are ready with the vaccines. We know how to make them. So Kevin says that because the genome sequence, can we just synthesize the S protein in vitro? And yeah. So Kevin, that is what I discussed. That we can make S proteins by recombinant. They can use them. Yes, definitely. They have to try it on human, Hussain. The reason for that is that we do not know that if we just inject the S proteins, what will happen to our body. When we inject the vaccine has some adjuvants in it, what will that do to our bodies? For example, I'll give you an example why a vaccine can become dangerous. There was a time that we made a vaccine for, I forgot the disease, but that vaccine caused our immune system to become so trained that when the actual pathogen came in, it attacked it with, our immune system became so active that almost like an allergic reaction, it started causing even more disease by inflammation. So imagine if you give me a vaccine today for SARS-CoV-2, that vaccine primes my immune system in such a large quantity that when the actual virus comes in, my whole immune system becomes active. That immune system is going to kill me. So they have to make vaccines in a way that sufficient amount of immune system is primed or trained, and when the actual pathogen comes in, the immune system is not going to either not work at all or work too much and cause damage. So these are the kind of things that they have to see. They have to see if it works or not. They have to see it doesn't work too much. They have to see that it doesn't cause any other harm. They have to see it doesn't interact with any other drugs. There are so many things to be tested. So that is about the vaccination. Absolutely. So don't trip is saying that it may be a risky strategy, but we may have to do it. Yeah. So it is possible that we would have to start the human trials very early and start working with them. So there is a question by Louis, will this different types of vaccine when applied on humans require different times to provoke? Of course, every vaccine may have a slightly different way of working, but the point is this, that if I'm not sick today and you give me vaccine, that gives me 10, 15 days of a chance to develop immunity. And then when the actual virus comes in, I can be ready. It is possible that you can give me passive immunization with that if I am on front lines, for example, if I'm a hospital care worker, and then while my vaccine is working, we can also have passive immunity. So by this technique, the cell will continue to produce S protein as messenger RNA remains there. This absolutely. But the more important thing is that as soon as the S protein is produced and our messenger RNA is degraded within a couple of days. So when the S proteins are produced, our immune system becomes trained. In the future, when the actual virus comes in, it would attack it. S protein itself does not cause much damage. So the conjugated or unconjugated vaccine simply means that we have a vaccine that by itself cannot cause enough reaction. So we create a conjugation of it so that the molecule becomes a little bigger so that there is a reaction. So nothing special in there. So I wish there was an S protein antagonist denaturing the spike protein. Absolutely. So one strategy for treatment is that they're trying to find, can we build something that would connect with S protein and just bind with it? Maybe immunoglobulins. So that is another strategy for vaccine, that you inject the immunoglobulins against the S protein, and as the virus comes in, immunoglobulins just connect with the S protein. That is exactly what our immune system will do when we do all these vaccinations. Guys, so thank you very much for your time. Tomorrow I want to do a special lecture on the latest updates on the treatment. There are so many new ways of treatment. There is a issue with the ventilators that they may be causing issues. Hydroxychloroquine has become a little bit more available, or there are more studies on that. Then the other drugs, there are some more drugs that are coming in. So I wanted to discuss what is the latest in the treatment of the SARS-CoV-2 or COVID-19. So tomorrow we'll have that discussion. So there's a question, if the virus attaches to ACE receptors, are patients who take ACE inhibitors? No, so I have done a lecture on that the American College of Cardiology has said that there is no data available that says that the people who are taking ARB or ACE-IS are at any additional risk. So I've done a lecture before as well. Plus, if you just Google American College of Cardiology with the ACE-I inhibitors and SARS-CoV, you'll be able to come to their study. All right, guys. So tomorrow we'll talk about the latest techniques or latest material available for the treatment of the SARS-CoV-2. I hope this is good. Thank you very much for joining. Please, please, please share this with your friends. That is the way to pay me back. I will be grateful for this. Thank you very much. Have a good day, guys. Thank you. Thank you.